Want quick marks in your entrance exams? Let's learn PCR. PCR is polymerase chain reaction. It is an in vitro method to amplify a specific DNA segment from one copy to billions. It was invented by Kary Mullis in 1985. It is a fast, accurate, and completely automated process. For a PCR, we need four things: the DNA with the targeted segment, two primers in excess amounts, the 4 DNTPs in excess amounts, and the heat stable DNA polymerase, that is Taq polymerase, from the bacterium Thermus aquaticus, and magnesium ions as cofactors for Taq polymerase. PCR takes place inside a thermal cyclone. PCR is a three-step process. First is denaturation. At about 90 to 98 degrees Celsius, the two strands of the double-stranded DNA separate. Step 2 is annealing. Cooling to about 40 to 60 degrees Celsius, the primers hybridize with the complementary sequences on the DNA strands. Step 3 is extension. At about 70 to 75 degrees Celsius, the Taq polymerase extends the primers and synthesizes new DNA strands. Each cycle takes about 3 to 4 minutes and doubles the amount of DNA. So there's exponential amplification. After amplification, the DNA is purified, sequenced, or ligated into a vector for cloning. So here's a bonus mnemonic to remember the sequence of steps and temperatures in PCR. First is distinction melts DNA. Denaturation happens at the high temperature of 90 to 98 degrees Celsius. Second is average primers attach. Annealing happens at 40 to 60 degrees Celsius, where primers hybridize with the complementary sequences on the strands. Third is extension, and for extension, remember, extend to fulfill. At about 70 to 75 degrees Celsius, the Taq polymerase extends the primers to synthesize new DNA strands. PCR made simple. Remember the steps, remember the temperatures, and score better in your entrance exams.